Hello, we're back online, and I'm very happy to announce the next talk, which is very relevant to the things that we discuss in this section. Particularly, we should always remember why we are looking for regulatory elements and for gene regulation. We want to understand how cells change, how different cell subtypes are determined by gene regulatory programs. And in this sense, today we have a very strong technology in terms of single-cell transcriptomics, which allows us to track different cells and different cell subpopulations. And to this end, we need tools, not only computational tools, but also real wet lab tools. And Anna Tarasevich from Helicon will present some news in microdissection in the context of transcriptomic studies and single-cell research. Anna, that's your turn. Please start.
So, may I start? Yes. Good afternoon, good evening, dear colleagues. It's great to be part of this online conference. And today, a bit news from the field of microdissection. Trying to switch slide. Great. So, to people who are not familiar with this technology, laser microdissection is a way of isolating tissue regions, single cells, or even small compartments like chromosomes or nuclei from a tissue section with a laser beam. So here on the slide, we can see the result of microdissection from a prostate cancer FFPE sample. So these regions marked with a star are the microdissected regions. So what can we do with microdissection? First of all, yes, releasing tissue sections. For example, if you want to separate tumor from normal tissue. We can cut out single cells. For example, if we are working with cell smears from liquid biopsy samples, so we can capture circulating tumor cells. We can capture single cells, for example, from the frozen brain tissue, for example, neurons, and work with this single cells on downstream analysis. Another application is forensics, where microdissection really helps to distinguish, for example, male cells from female cells. It's a very important tool to solve some mixed cases of sexual assault. So it has a really broad application range. Another application is isolation of single living cells for recultivation, or even isolating of parts of cells, so chromosomes, nuclei, or something else. If we talk about downstream applications, they are really broad. So we can do either gene expression analysis, all kinds of proteomics, mutation analysis, all you can think of. So there are several types of microdissection. There are two main types. The first one uses the UV laser, ultraviolet. The second one, the IR laser, so the infrared laser. Here the two pictures on the left show the UV laser technology. So it's based on cutting. So you see here the some membrane slide here marked blue, and the tissue region marked green, which we want to isolate. So we focus the laser on the tissue, and as a result, the microdissected tissue falls, either it falls down to a transfer vessel, or it is catapulated up upwards to a transfer vessel. This is one thing. The other technology is based on the IR laser, and here we use a special adhesive cap, which has here marked red, which has a transparent film on it. And this film, when it is activated by the heat of the IR laser, attaches to the tissue of interest. And then we just lift the cap and lift the tissue. Both of these methods have both advantages and drawbacks. So with the laser microdissection, with the UV laser, which is very strong, we can cut large areas of tissue or hard tissues like bones very fast. And so we also can cut live cells in petri dishes. But the drawback is that the UV light can destruct some nucleic acids or biomolecules near their cutting edge. This is why it's not very good suitable for single cell isolation. And special membrane slides are needed, which are expensive. If talking about the IR laser, this is a gentle laser, and it doesn't disturb the integrity of biomolecules. This is why we can work with single cells, and this technology works with all types of slides. But there are some drawbacks, of course. It's the speed, it's slower, and we have a limitation of the tissue thickness. Here are some proof of principle results which show that really the UV laser, so here we see a UV laser cut and an IR laser cut. And we see that single-stranded DNA, so the disturbed DNA is present only at the edge of the UV laser. So really the IR laser is more gentle, and it doesn't disrupt the tissue. This is another proof of principle experiment where single cells were captured, and we see that after the IR laser capture, we have a much better RNA quality in downstream analysis. But as we see, both cutting methods have their advantages, and our partner and really great technological company Fluidigm has developed a new system, which is called Aculift, which is now the only commercially available platform which has both the IR and the UV cutting laser. Important to mention is that it was developed with in collaboration with Dr. Lance Leota, who is the key inventor of the LCM technology itself. So he invented this infrared laser. So in this system, we can profit from advantages from both of the lasers: hard tissues and fast cutting, live cell cutting, and single cell analysis. So really, there's a huge number of downstream applications. This is how we actually a screenshot of the program where we can see how we select our target areas. So with the this red spots are the spots where our infrared laser attaches to the tissue, and the violet circles, these are the lines where the UV laser cuts. So we can lift our tissue from the slide via this attachment points of the IR laser. This is a video I wanted to show about the system itself. It's very cute, I think. Do you hear some music or not? So this is how it works. And this is my final slide, just to show the differences of the Aculift system from all the other systems on the market. So it has both lasers, the IR and the UV laser. It can work with single cells as well as with large areas. One important thing is that it really is optimized, is a very optimized system. This is why the price is very nice too. So it's twice the cheap as the previous models of similar systems. And yeah, it's very flexible. Can use any types of slides and any downstream chemistry. So if you are interested, if laser microdissection is of interest to you, then we can organize a virtual demo. We can always cooperate and yeah, let's see if this system really finds its place on the market. Well, thanks a lot for listening. If some questions are present, I'll be glad to answer.
Anna, thank you so much. Actually, I believe many or most of the people who are listening to us now are computational biologists or purely dry lab people. And I think it's really important for us to have a look at what actually happens in the wet lab. So the numbers and the gene expression that we are getting are not just some magic coming out of the black box. There are real cells and the real technology behind it. So I think it's very important for us, but I'm not sure if we can truly ask proper questions regarding suitability of such system for, let's say, analysis that's interesting in terms of systems biology. Of course, we want single cells, we want data on gene expression and proteomics from single cells. And I hope we can transfer this knowledge that such systems exist and are available for demonstration for our wet lab partners. So thank you again. And we don't have any questions from chat. So I think we move on and switch to the next talk. Thank you so much.
Thanks a lot.